The purpose of this presentation is to describe an overview of chapters 12 and 13, the cell cycle, and the phases mitosis and meiosis. Presented by Olson and Taylor Kelly. Let's begin by discussing an integral part of the cell cycle and the life of a cell division and cellular organization. From parent cell into two daughter or sister cells, which is a process of passing identical cell DNA, or genetic material known as genome, to its cellular offspring. This, being the crucial function of cell division. Did you know that the DNA-packed structures, known as chromosomes, got their name thanks to Walter Fleming, a German anatomist who developed color dyes in 1882. Hence, the term, chroma, which means color. These dyes allowed the relationship behaviors between the short mitotic phase, which alternate with the larger stage, known as interphase, became observable for the very first time. This larger phase is made up by three parts. So, what are these three parts? The G1 phase, or first gap, the S phase, or synthesis phase, and the G2 phase, or second gap. The G phases were misnamed because when first observed, the cells appeared as inactive. However, we now know otherwise, as a cell grows by producing proteins and cytoplasmic organelles such as mitochondria and endoplasmic reticulum. Let's explore the unique characteristics and parts of the 6 phases of mitosis, which is the division of genetic material in the nucleus. G2 of interphase. Key features. A nuclear envelope encloses the nucleus, which contains one or more nucleoli. 2 centrosomes have formed, by duplicating from their initial single form. Chromosomes, duplicated during the S phase, cannot be seen individually, as they have not yet condensed. Prophase. Key features. Stage 1 of mitosis, the chromatin fibers become more tightly coiled, condensing into discrete chromosomes which can be seen with a light microscope. The nucleoli disappear, and the mitotic spindle forms. Prometaphase. Key features. The second stage of mitosis, in which the nuclear envelope fragments, and the spindle microtubules attach to the kinetochores of the chromosomes. Metaphase. Key features. The third stage of mitosis, in which the spindle is complete. The chromosomes, attached to microtubules at their kinetochores, are all aligned at the metaphase plate. Anaphase. Key features. The fourth stage of mitosis, in which the chromatids of each chromosome have separated, and the daughter chromosomes are moving to the poles of the cell in a fast jerking motion, which causes the ends of the chromosomes to lag back towards the metaphase plate. Telophase and cytokinesis. Key features. Telophase is considered the fifth stage and final stage of mitosis, in which daughter nuclei are forming, and cytokinesis, named the sixth phase, above the latter, finishes the stages of mitosis, in which the formation of the cleavage burrow is started and finished, pinching the cell in two. In conclusion, mitosis can be summarized by simply saying that it is a process which takes place in the nucleus of a dividing cell, and typically involves a series of consecutive stages consisting of prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis, which results in the formation of 2 new nuclei, each having the same number of chromosomes as the parent nucleus. Now, let's explore the unique characteristics and phases of meiosis. Several steps of meiosis closely resemble corresponding steps in mitosis. Meiosis, like mitosis, is preceded by the duplication of chromosomes, but significantly, this single duplication is followed by not one, but 2 consecutive cell divisions called meiosis I and meiosis II. This double cell division results in 4 daughter cells instead of the 2 daughter cells of mitosis. However, each of meiosis' 4 daughter cells have only half as many chromosomes as the parent cell, 1 set instead of mitosis's 2. Meiosis stage 1. The process of separating homologous chromosomes. Key features. Meiosis begins with the prophase I, which includes movement of the centrosome, as well as spindle formation and nuclear envelope breakdown occur in mitosis. Chromosomes condense rapidly. During early prophase I, each chromosome pairs with its homologue, aligned gene by gene, and crossing over or the exchange of genetic material between homologues. The X-shaped, at regions, are called chiasmata where crossovers have occurred. Metaphase 1. Key features. Pairs of homologous chromosomes are now arranged at the metaphase plate, with one chromosome of each pair facing each pole. Both chromatids of one homolog are attached to kinetochore microtubules from one pole. The chromatids of the other homolog are attached to microtubules from the opposite pole. Anaphase 1. Key features. In the third phase of meiosis, homologs separate and move to opposite poles which are guided by the spindle apparatus sister chromatids. Cohesion persists at the centromere, causing chromatids to move as a unit toward the same pole. Telophase 1 and cytokinesis. Key features. When telophase 1 begins, each half of the cell has a complete haploid set of duplicated chromosomes. Each chromosome is composed of two sister chromatids. One or both chromatids include regions of non-sister chromatid DNA. Cytokinesis. Division of the cytoplasm usually occurs at the same time as telophase 1, forming two haploid daughter cells characterized by a cleavage furrow. Meiosis stage 2. The process of separating sister chromatids. This stage begins with prophase 2. Key features. A spindle apparatus forms, and in late prophase 2, chromosomes which are each still composed of two chromatids associated at the centromere, are moved by microtubules toward the metaphase 2 plate. Metaphase 2. Key features. Chromosomes are positioned at the metaphase plate, as in mitosis. Also, because of crossing over, the two sister chromatids of each chromosome are not genetically identical. Anaphase 2. Key features. The breakdown of proteins holding the sister chromatids together at the centromere allows the chromatid to separate. The chromatids move toward opposite poles, as individual chromosomes. Telophase 2 and cytokinesis. Key features. The nuclei form the chromosome begin to condensing, and cytokinesis occurs. The mitotic division of one parent cell produces four daughter cells, each with a haploid set of chromosomes that are genetically distinct from one another. In simple terms, you can simplify meiosis down to its importance, which is the genetic variation of sexual reproduction.